Photosynthesis is divided into light-dependent reaction and light-independent reaction. Light-dependent reaction requires energy input from the sunlight or photons to split water into oxygen, produces ATP energy as well as reducing agent called NADPH. Oxygen gas is the byproduct and will be removed from the leaf through the stoma. ATP and NADPH will be used in the light-independent reaction, or also known as the Calvin cycle, to fix carbon dioxide into simple sugar. The light-dependent reaction takes place on the thylakoid membrane of the chloroplast, while the Calvin cycle takes place inside the stroma of the chloroplast. First, we will discuss the processes involved in the light-dependent reaction. Light-dependent reaction starts with photoactivation of both photosystem 1 and photosystem 2. During photoactivation, photons will be absorbed by the antenna pigments of the photosystem. The energy will be transferred from one antenna pigment to another until it reaches the reaction center chlorophyll. In photosystem 2, the reaction center chlorophyll is known as P680. While in photosystem 1, the reaction center chlorophyll is P700. Absorption of energy from the antenna pigments leads to excitation of electron in the reaction center chlorophyll A. This is due to the electron having higher energy level because they have absorbed the energy transferred from the antenna pigments. The excited electron will be captured by the primary electron acceptor. This means that P680 will undergo oxidation and now become P680+, or is also known as electron deficient P680. Similar to P680, absorption of energy from the antenna pigment by P700 will also cause excitation of electron from P700, followed by the excited electron being captured by the primary electron acceptor, and the P700 is oxidized into P700+, or electron deficient P700. Overall, we can see that photons or energy from the light is being used to excite electron from P680 or the P700 to a higher energy state so that it will be captured by the primary electron acceptor. As a result, both P680 and the P700 will become oxidized into P680+ or P700+. Next is photolysis of water, during which water molecule will be split into oxygen, proton, and electron. The electron is then used to replace the electron previously lost during photoactivation of photosystem 2. P680+ turn back into P680. The next process in light-dependent reaction is photophosphorylation. Photophosphorylation can occur by two means. The first one is through non-cyclic flow of electron, and the second one is through cyclic flow of electron. During non-cyclic photophosphorylation, there are two products. The first one is ATP, the second one is NADPH. This non-cyclic flow of electron occurs most of the time during light-dependent reaction. The non-cyclic flow of electron starts with the electron from the primary electron acceptor of PS2 is transferred to plastoquinone and then to cytochrome complex and lastly to plastocyanine. The energy released as the electron is passed through the cytochrome complex is used to generate the potential energy, which is then used to phosphorylate ADP into ATP by chemiosmosis. After that, plastocyanine will transfer the electron to P700+ of the photosystem 1. This is the point where the electron previously lost from the P700 is replaced with excited electron from P680 of the photosystem 2. Doing so will reduce P700+ back to P700. In the next step, the excited electron captured from the P700 will be transferred to another mobile carrier molecule called ferredoxin. Ferredoxin will transfer the electron to an enzyme called NADP+ reductase, which uses the electron to reduce NADP+ into NADPH. This shows that the final electron acceptor for non-cyclic photophosphorylation is NADP+. In addition, the transfer of electron from ferredoxin to NADP+ reductase does not involve cytochrome complex, which means that there is no production of ATP by chemiosmosis. Only the flow of electron from photosystem 2 to the P700+ will involve generation of ATP by chemiosmosis. The flow of electron from PS1 to the NADP+ reductase does not involve production of ATP by chemiosmosis. This is light-dependent reaction with non-cyclic photophosphorylation. Next is light-dependent reaction with cyclic photophosphorylation. Cyclic photophosphorylation will only involve PS1 and not PS2. The electron captured by the primary electron acceptor of the photosystem 1 will be transferred to ferredoxin and then to the cytochrome complex instead of to the NADP+ reductase as we seen in the non-cyclic photophosphorylation. From cytochrome complex, the electron will be passed to plastocyanin and then eventually to P700+. Similar to what happened previously during non-cyclic photophosphorylation, flow of electron through the cytochrome complex will generate potential energy, which is then used to phosphorylate ADP into ATP during chemiosmosis, and then P700+ will be reduced to P700. Because of the cyclic flow of electron from P700 through cytochrome complex and then back to P700, this is why it is known as the cyclic photophosphorylation. The only product for cyclic photophosphorylation is ATP. This is very important because the light-independent reaction or the Calvin cycle requires more ATP than NADPH. Another important point to take note is that light-dependent reaction with cyclic photophosphorylation does not involve photolysis of water, since it does not involve photosystem 2. The electron loss during photoactivation of P700 will be replaced with the same electron. So the final electron acceptor is P700+.